When cleaning limits are established, they are calculated using a defined exposure scenario derived from a toxicological boundary. That toxicological boundary, such as an HBEL or PDE, remains constant and it defines the level of exposure considered safe for patients. This is what we call the exposure scenario. When equipment is dedicated and product sequencing is fixed, the exposure scenario remains stable. However, in shared equipment environments, the next manufactured product is not always the same after cleaning. Different products may be scheduled depending on planning, demand, or batch sequencing. As soon as more than one next product becomes possible, you are no longer managing a single defined transition. Instead of one scenario, such as product A to product B, you may instead be managing several plausible transitions, such as product A to B, then product A to C, or product A to D. Each transition may involve different batch sizes, dosing regimens, routes of administration, or patient populations, all of which affect how the toxicological boundary is converted into an operational cleaning limit. The toxicological boundary itself remains unchanged, but the resulting cleaning limit may vary depending on the receiving product. That variability must be governed.